All right, this is the podcast for AP Biology. This is on the nervous system, chapter 34. In the first section, we're going to learn about neurons and glial cells. And those are the two types of cells in the nervous system. Neurons, which are nerve cells, and glia, which are the glial cells. All right, first we're going to learn about neurons. There are four main parts of a neuron: the cell body, the dendrite, the axon, and the axon terminal. We will go through each one of these. All right, the cell body is first. The cell body is like the cell. It contains the nucleus and all the different organelles. But when you usually see a diagram of the cell body, you're not going to be able to see any organelles except the nucleus. The cell body has dendrites extending from it, which we will see what that is in a minute. All right, the dendrites extend from the cell body. They're shrub-like. They're like little lines sticking out. They bring information from other neurons or sensory cells to the cell body. There's a different degree of branching, so it can get more complicated or simple depending on the type of neuron it is. All right, third part of a neuron is the axon. The axon is the long projection off the cell body. It can be pretty long. It can extend, for example, from our spinal cord all the way down to our toes. They're like the telephone lines of the nervous system. They act on information that is received by the dendrites. And the axon is what is generating an action potential. An action potential is the nerve impulse. That action potential, or the nerve impulse, will travel down the axon toward a target cell. A whole bunch of axons bundled together is called a nerve. All right, the last part of the neuron is called the axon terminal. So we have this axon going down and the really long extension. At the end of those nerve endings is the axon terminal. So it's basically a swelling at the end. It is very close to the membrane of the target cell where that action impulse is traveling to. And what it does is that it forms a synapse. A synapse is just a basically tiny little gap in between the two neurons, between the neuron 1 and maybe the target neuron. And so the tiny gap across which two neurons are communicating, those two neurons that are communicating are either communicating through electrical signals or with chemical signals. The neuron that's sending the information down the axon to the axon terminal is called the presynaptic neuron, and the neuron that is receiving the information is the postsynaptic neuron. All right, so this diagram is in your book in chapter 34. It's figure 34.1. And so on your diagram, please label the 5 parts. The dendrites are the little branches that are on the cell body. In the cell body, in this diagram, you can kind of see you can see the nucleus and the ER in there. So that is the main cell. And then the axon is the long extension coming down. And then you can see the axon ends in these little swellings at the end of those tips is called the axon terminal. And then you can see another neuron down below. The axon hillock is part of the cell body and part of the axon, where the axon is connected to. So where the axon is connected to at the cell body, that is called the axon hillock. And then please make sure you have the arrow of the direction of the action potential or the nerve impulse. It travels from the cell body of the presynaptic cell or neuron to the postsynaptic cell. So it travels down the axon. All right, the second type of cell that's in the nervous system are the glial cells. And there's way more glial cells than there are neurons. So they're more numerous. They have basically 3 jobs. The first job is they release neurotransmitters. Neurotransmitters are chemicals associated with nerve impulses, and we'll see their role in a little bit. A second function of the glial cells is they provide homeostasis for neurons, because there's going to be these neurotransmitters released out of the axon terminals, and these glial cells, which are releasing these neurotransmitters, are actually going to clear them out as well. So they provide homeostasis for neurons by clearing the synapse of neurotransmitters. So you will see that also in a little bit. The third function of the glial cells is to repair neurons and remove any neurons that are not working or dead neurons. There are 3 different kinds of glial cells: astrocytes, microglia, and Schwann cells. One thing, keep in mind the Schwann cells is spelt wrong. There's a C missing. It should be SCHWANN. The first type of glial cell is an astrocyte. These are found in the brain. They surround the smallest, most permeable blood vessels in the brain. They contribute to the blood-brain barrier. That is a barrier that prevents toxic chemicals from reaching the brain. This blood-brain barrier usually prevents antibodies from entering the brain as well. So the brain gets its immune defenses by another glial cell, our next one, called microglia. This one is pretty simple. The second type of glial cell is the microglia. It is a glial cell that acts as a macrophage, and it is a mediator of the inflammatory response in the brain. All right, the third type of glial cell is the Schwann cells. These cells form a multi-layered wrap on axons. So the axon that's coming down is covered with these Schwann cells. It forms a lipid-rich sheath called myelin. Myelinated axons have a white appearance, giving rise to white matter in the brain. Areas of the brain that do not appear white, there are areas that are rich in cell bodies, are actually called gray matter. There are spaces between the Schwann cells. Those spaces are called the nodes of Ranvier. And if you, in regular bio, watched Lorenzo's oil, ALD, which is what Lorenzo had, and multiple sclerosis are diseases where this myelin is affected. All right, so this diagram is in your book. It's figure 34.2, part 1. And so you can see the darker purple, or the pinkish purple, is the axon in the middle there. And then the purple around it are all those myelin cells, which they have lipids in them. And so those are the Schwann cells. And then you can see the spaces between them. And so those spaces are the nodes of Ranvier. And these are regular cells that have a nucleus, and that's what that other thing is pointing to. All right, so looking at what neurons do, there are 3 function categories of neurons, or 3 types of neurons: afferent neurons, efferent neurons, and interneurons. Afferent neurons carry information into the nervous system coming from a sensory cell, and that it transduces the sensory stimuli into action potentials. So it's carrying sensory information and it's making it into a nerve impulse or an action and potential. An efferent neuron carries commands to physiological and behavioral effectors such as muscles and glands. For example, a motor neuron would carry commands to a muscle cell. And then you have interneurons. And these are the ones that are found in the brain. Most neurons are in the brain are interneurons. They integrate and store information, and they communicate between afferent and efferent neurons. All right, there are 3 types of neural networks that I want to show you. The very first one, or it's really basic one, is one that would be found in like a sea anemone. That's called a nerve net. And so you can see in a sea anemone how it's net-like. And it serves, though, for simple behaviors, such as contraction and relaxation. So that would be a nerve net. And then you can get more and have actual ganglia, which is like a mini brain. And so an earthworm, for example, he has an anterior ganglia, and they have ganglia in their ventral nerve cord. And so they have more than one ganglia and a whole bunch of nerves that run through the whole earthworm. So there are ganglia in each segment. You can see that in the picture. That coordinates movement, and the anterior ganglia controls more complex behavior. And then thirdly, you have the brain. And so this would be more complex, like a human, where you have the central nervous system, where the human brain and the spinal cord are working together. And then you have the peripheral nervous system, which is when everything's being communicated with the cells and the organs of the entire body. All right, the second section of this chapter, 34.2, is on the electrical signals. All right, so action potentials. Action potential is just a nerve impulse. It's just a scientific term for that. What action potentials are and what they do is they carry information along these neurons. And what happens is there's a sudden and large change in the membrane potential. This is a difference in electrical charge across the plasma membrane. That change in the membrane potential is traveling along the axon of the neuron, and it causes a release of chemical signals at the axon terminal. And remember, these chemicals are the neurotransmitters. All right, another term you need to know is voltage. A voltage is a measure of the difference in the electrical charges between two different points. It represents potential energy because opposite charges will move together if given a chance. In wires, electrical current is carried by electrons. However, we're talking about membranes. So in solutions and across cell membranes, the electric current is carried by ions. So let's look at what ions are involved. All right, the ions that are involved in an action potential, there are 4 of them. You have sodium, potassium, calcium, and chloride. And their symbols and charge is also listed there for you. In cells, all of these ions are kept at different concentrations, inside and outside of the cell. The result of these differences in concentrations is the voltage across the cell membrane. And this difference is called the membrane potential. All right, a resting membrane potential occurs in an inactive neuron. An inactive neuron is a neuron that is not sending a signal and is not receiving a signal. A resting membrane potential is typically between -60 and -70 millivolts, which is abbreviated MV. The minus sign refers to an electrically negative cell compared to the outside of the cell. These action potentials, or nerve impulses, are generated when there's a sudden change in this voltage to where it is more positive inside than outside. And so how is this done? How is an action potential done? Is the sodium-potassium pump. All right, so as a review, let's review the sodium-potassium pump. Remember, it is a form of active transport, so it uses ATP, adenosine triphosphate. And in the sodium-potassium pump, we have sodium ions being pumped out of the cell in exchange for potassium ions from the outside of the cell. Remember, this exchange is uneven. The sodium-potassium pump is constantly pumping sodium out and potassium, but the concentration of sodium is actually higher outside than inside, and the concentration of potassium is higher inside than outside. These concentration gradients will be used to generate the resting potential and changes in the resting potential. So how would the resting potential change? All right, so here's a diagram of the sodium-potassium pump. And so we're breaking down ATP. Here is our protein in the cell membrane. And we can see we have 3 sodium ions being pumped out of the cell. And potassium ions are being pumped in. Okay, so what's occurring here, the sodium-potassium pump, is as sodium ions are being pumped out of the cell, sodium ions are represented by the little yellow circles. You can see we're getting tons of sodium out here, which have a positive charge. And then potassium ions are being pumped into the cell. So you can see there's lots and lots of potassium ions into the cell. Okay? And they both have a positive charge. However, remember, there are more sodium ions being pumped out of the cell than there are potassium ions being pumped into the cell. So overall, there is more of a positive charge on the outside of the cell than on the inside of the cell. In comparison, the inside of the cell is more negative and the outside of the cell is more positive. And so this is where we are starting at. All right, so let's say a stimulus occurs. Let's say light hit something. You get pinched, whatever that is. That is going to trigger a voltage-gated sodium channel to open up. And that is going to bring sodium ions into the cell. So it's going in a backwards direction. Sodium is going to go inside the cell because sodium will move from high to low. And there is more sodium right now outside the cell than inside the cell. So inside of the cell is becoming less and less negative. This is called depolarization, because we're going to go, let's say, from -70 millivolts to -50 millivolts. When the inside of the neuron becomes less negative, it means it's more positive. All right, so what happens next is as sodium gets pulled in, additional voltage-gated sodium channels are going to open. That causes a rapid spike of depolarization, and that is what an action potential is. And remember, the action potential is going to actually travel down the axon. The depolarization is going to trigger voltage-gated potassium ions to open, and that is going to allow potassium ions to flow out of the cell from high to low. This is called hyperpolarization. Or the membrane is hyperpolarized, which means the membrane is becoming even more negative. All right, so let's say the stimulus hit the neuron. Right away, the sodium channels are going to open up, and sodium is going to flush into the axon. And so you can see there's a change in the membrane polarity right here, where the inside of the cell is going to become more positive and the outside of the cell is going to change to negative. And you can see that flow of change, or the action potential, is going to travel down the axon. And you can see that here as well, that the action potential is traveling down, and you can see the change in the charge. That is the depolarization, which causes the action potential. All right, some more on action potentials. The strength of the signal doesn't change during the travel down the axon. It is an all or nothing response. There's a positive feedback mechanism to ensure that the action potentials will always rise to their maximum value, or nothing will happen. It is also self-regenerating. One action potential will stimulate another. It cannot go in reverse. It can't go the opposite direction due to the refractory period. A refractory period is the time in which the membrane is returning back to resting potential. You can see that on the diagram I just showed you, that the charge goes back to normal. It will travel faster in axons that have that myelin around it, or if the axon is larger. So squids, their axons are very big in diameter, and so their response time is very rapid. All right, communication between neurons. Once an action potential reaches the axon terminal, it will release neurotransmitters, these are chemicals, into the synaptic cleft. The synaptic cleft is that space, that gap that exists between the neurons. These neurotransmitters are going to bind to receptor proteins, and they're going to open the ion channels of the new neuron cell. If enough ion channels are opened, the action potential will continue to the next neuron. If not enough ion channels open up, then the nervous signal will be terminated. After the neurotransmitters have opened the ion channels, they will be cleared out of the synaptic cleft by being reabsorbed by the neuron that released them, or broken down by enzymes. All right, so here is the action potential traveling down the axon. All the neurotransmitters are these little red dots, and they're inside these vesicles. And so as the action potential reaches the end of the axon terminal, and keep in mind that this one is the presynaptic neuron, and this one here is the postsynaptic neuron. So as the action potential travels down the axon and reaches the end, that triggers the release of neurotransmitters. So you can see the vesicle fuses in with the membrane, and these neurotransmitters leave the neuron, and they go into the cleft, which is the gap. All right, so those neurotransmitters are going to bind to a receptor that are on these proteins, or ion channels, and if enough of these bindings occur, these ion channels will open up, and the neurotransmitter will go in, and the action potential can actually continue on. Otherwise, if that doesn't occur and not enough of these open up, then these neurotransmitters will be cleared out by enzymes, for example, and the action potential will stop. So this is the details for the chemical synapses. Neurotransmitters released from a presynaptic cell are going to bind to receptors in the membrane of the postsynaptic cell. I just showed you that on the diagram. The neurotransmitter used by all vertebrates during this neuromuscular synapsis is acetylcholine, and that is abbreviated ACH. There are 7 steps to a chemical synapsis. So these 7 steps are just basically giving you more detail on what I went through on the diagram. So step number 1, the action potential is going to arrive at the axon terminal. Step number 2, sodium channels are going to open. Depolarization is going to cause the voltage-gated calcium ion channels to open up. Step 3, calcium is going to enter the cell, and that is what triggers the fusion of these vesicles that have the neurotransmitter. In this case, we're talking about acetylcholine with the presynaptic membrane. Step 4, the acetylcholine or neurotransmitter molecules are going to diffuse across the synaptic cleft, and they're going to bind to those receptors that are on the postsynaptic membrane. Number 5, when binding occurs, they open up their channels and they will depolarize the postsynaptic membrane. Number 6, the spreading depolarization fires an action potential in the postsynaptic membrane, and then any acetylcholine that is left in the synaptic cleft will be broken down. So this diagram is just a little more in depth, showing the 7 steps. Your action potential is traveling down the axon. That's step number 1. Step number 2, these sodium ions are opening up because the action potential is coming down. But what happens is that in the axon terminal, that's going to cause these calcium channels to open up. And so calcium gets flooded into the axon terminal. And so that enters the cell, and that just triggers these vesicles to fuse with the membrane. And you can see all the acetylcholine neurotransmitters are being released into the cleft. All right, these acetylcholine molecules are going to diffuse across the synaptic cleft, and they're going to bind to these receptors. You can see that on these receptor proteins on the postsynaptic cell. And then if enough of those open up, these channels will open, and the acetylcholine will go in, and it will depolarize, change the polarity of the postsynaptic membrane. And that fires the action potential to continue on. Any acetylcholine that is left over will be cleared out and recycled. All right, so neurotransmitters, action, how does it actually stop? It has to be cleared from the synapses. That's how they maintain homeostasis. Enzymes may destroy the neurotransmitters, or neurotransmitters might just simply diffuse away from the synaptic cleft. There are so many different types of neurotransmitters, and you might have heard of some of these before, but there are more than 50 that are recognized. We don't go into detail on them, but acetylcholine is one that's used in the brain with motor neurons. Other ones you might have heard of before are GABA, dopamine, norepinephrine, which we talked about before, serotonin, and endorphins. These are all neurotransmitters. All right, just a side note is that drugs can interfere with neurotransmitter release. There's a bacteria called Clostridium, and it releases these toxins, and these toxins will destroy proteins that are necessary for that binding of the vesicles to the presynaptic membrane. So these toxins cause botulism and tetanus, and it's a fatal disease because it involves muscle impairment due to the loss of neurotransmitters being able to be released because the vesicles can't bind. And that finishes our notes for chapter 34.